Greetings, everybody. This is Mason Brooks from Active Motif, and I'm welcoming you to this great webinar presentation that we've put together entitled CUT&Tag versus ChIP-Seq from Data Generation to Analysis. This is a joint webinar between Active Motif and our colleagues at Basepair. The first part of this presentation will be given by Active Motif's Dr. Sarantis Chlamydas, who's a Senior Market Development Manager and overall epigenetics expert. He'll be discussing some recently developed advanced epigenetics techniques like CUT&Tag and ChIP-Seq and how they can be applied to your research projects. Before we begin, if you have any questions, please use the Q&A function at the bottom of your screens to post questions, and we'll try to answer as many as possible at the end of the presentation, or we'll follow up after the webinar concludes by email. And so now on with the presentations. Thanks for joining us, Dr. Chlamydas.
Thank you very much, Mason. I'm really happy to give this presentation to you. I will discuss today about ChIPSeq and CUT&Tag tools that we have developed internally in Active Motif. First, an introduction about our capabilities. We are focusing mainly in epigenetics and chromatin biology. Our tools are focused in epigenetic discovery and targeted epigenetics. We provide end-to-end services for ChIPSeq, RIME, CUT&Tag, ModSpec, High-C, and for DNA methylation, we have RBS and target bisulfite sequencing. Of course, in the level of single-cell biology, we have ATAC-Seq single-cell and RNA-Seq single-cell. Everything supported by bioinformatic analysis. In our long experience and experiments with epigenetics, we have dealt with a lot of variety of cells and tissues, from PBMCs to liver, brain, ovarian tissue. And this is an example in regards to the ATAC-Seq experiments. Today, I will give you a short introduction about epigenetics. I will spend a little bit of time explaining the ChIPSeq, CUT&Tag, and how they can be combined in the context of epigenetics, and some examples how you can apply the new technologies with the help of bioinformatic analysis in biomarker identification, validation, and translational medicine. Epigenetics actually is the mechanism where regulates and defines the phenotype of a certain cells. In all the cells in our actual organism, we have the same DNA, but the characteristics are coming from different epigenetic mechanisms. Here, they are depicted the main ones, that there are histone modifications and DNA methylation and DNA modifications. This is a really nice example to show how the epigenetics can influence and can define our phenotype. These twins, they have exactly the same DNA, they have difference in DNA methylation level. And these differences, they are accumulating during aging as well. This is a really excellent example how the epigenome defines our phenotypes. What it does, the epigenetics is taking the hardware, is the software that takes the hardware and put the phrases in the right way in order to make proper phrases. In the context of precision medicine, it's really important the integration of multiomics data, the integration of multiomics data. For sure, we have here talk about genomics, transcriptomics. Active Motif also introduced epiomics in the context of omics. Of course, really important, the strong bioinformatic analysis and the strong bioinformatic support in order to integrate the different omics data in order to provide solutions towards precision medicine. In regards to chromatin, the most common protocols that they are known so far is the ChIPSeq, chromatin immunoprecipitation, is a fixed base protocol. We need to have the sonication to cut the chromatin and specific antibody to pull down actually fragments of interest, and then sequencing to map the region's localization of malprotein of interest. The new version is the CUT&Tag, where we don't use a fixation. It's not a fixation-based protocol. It's a native conditions. We mobilize with concavalent beads cells and nuclei, and we use protein A fused to TN5 transposon to integrate actually sequencing adapters at the regions where my protein is located. And not last but not least, the ATAC-Seq is a non-antibody-based protocol where we use a transposase to integrate sequencing adapters at the open chromatin regions. At the end, we sequence the open chromatin regions. Active Motif offers a complete toolbox for all of these techniques, including kits, assays, but also end-to-end services. As I mentioned before, for ChIP and CUT&Tag are really important, the cutting, are really important to have the proper cutting in order to achieve high resolution mapping. Starting with ChIP, we are really happy because this year we launched the Pixel. It's a high precision multi-sample sonicator where we have tested and validated for different applications in the field of epigenetics, for different applications in the field of multiomics. We have tried DNA for applications like Medip-Seq, RNA for RNA-Seq, chromatin to prepare ChIPSeq and later on libraries for sequencing, and proteins from different tissues and cells, also FFP samples. It's a 9-6 multiplex sonicator. You just don't need to transfer your cells. You just grow your cells overnight in a simple coning plate, and then you perform next day the end-to-end experiment. You can run 12 unique sonication parameters at once. We take high reproducible searing. We don't need expensive consumables, and we have a series of assays that support the multiplex nature of the machine. Here's an example at the level of chromatin, how we use it from 100,000 cells to million cells. We don't use more than one buffer. We don't include nuclear isolation, and you can appreciate high precision sonication searing. There is an example that we try to compare, and we have all the data in our nucleic acid research paper, different commercial available sonicators. And as you can see with the Pixel, we take really consistency in our sonication, also during time. But we didn't test it only for cells. We tried different tissue types from mouse and human, kidney, liver, spleen, brain. We have seen that for human chromatin, we need for a complete set of 96 experiments, 72 minutes. For mouse chromatin, we will sonicate roughly 36 minutes. Here's an example how the sonication looks like. Here's an example actually from human brain. You can appreciate we take highly enrichment around 300 base pairs. Same valid also for mouse brain. And most of the chromatin, both mouse and human, is sonicated with a highly efficiency. Of course, doesn't say much. This just they don't say much. We tried also to run in parallel the ChIPSeq experiments with this chromatin. You can appreciate using histone modifications, in that case H3K9 acetylation, in different actually chromatin from different tissues. We take high reproducible data in both human and mice. That means that the quality of the chromatin that we take for sonication not only is high reproducible, but is super really nice tested and validated for ChIPSeq experiments. We try different cell lines with different histone marks. This is an experiment that we run in the same plate at the same time. We try cell lines that they are both attached and in suspension, with histone marks that they are for open chromatin regions like H3K4 mono and tri, but also for closed chromatin regions, H3K27 tri, and a transcription factor and insulator CTCN. As you can appreciate, we take a complete data with all the epigenomic profile of these cell lines. Changing a little bit gears, now we have the new entry. I will present you the protocol of CUT&Tag. Initially originated in the laboratory of Henikov. As I explained you before, you don't need to fix the cells. You just immobilize cells and nuclei on Coq-Avaline beads. You use a specific antibody to target the protein of interest, a secondary antibody, and then a fused protein A to TN5 that brings also the sequencing adapters. by activating the transposase, then you integrate the sequencing adapters at the region where the antibody is located. It's a non-formaldehyde cross-link protocol. You don't need sonication. We have seen that we take a high resolution for mapping the histone modifications and some transcription factors, less starting material, and of course, you save a lot of time and money since you don't spend time to generate libraries. It's happening at once with the use of the transposase. Some examples that they came originally from the paper of Henikov's lab. You can see really nice correlation with both closed chromatin marks, H3K27 tri, and open chromatin marks, H4K mono. Really nice ChIPSeq and CUTTag correlate for both of these marks. Here I try to summarize the pros and cons of these technologies, and I'm really happy to have your feedback on that as well. For ChIP is the golden standard traditional technique. We have a variety of samples tested and validated, including biopsies FFP samples. There are a lot of validated antibodies that also a lot of consortia have used and have tried. Number of publications and reference data are super high. And of course, there are normalization protocols like spiking in order to make it as quantifiable as possible. Like a cons, we require a lot of input material, and there are fixed conditions and sonication that may have some problems. They may create some problems for certain antibodies. On the other hand, CUT&Tag is working in native conditions. We have seen that we take better resolution. We need fewer amount of cells. Now people, they're talking about single cells also. We save time of preparing the library with the use of TM5. We go lower to the number of the sequencing, and I can show you that we need 8 million reads, for example, to take really nice reproducible data. But on cons, we have few publications, less validated antibodies in regards to transcription factors, for example, and of course, less sample types validated compared to the ChIPSeq. Here's an example where you can see for the same histone marks like H3K27 tri and K4 mono, we need less sequencing depth in comparison to ChIPSeq. For a ChIPSeq, we need like 50 million reads. For a CUT&Tag, we write 8 million reads to take really nice and reproducible data. Some examples from our laboratory using our kit. You will see that we run side by side different histone modifications and transcription factors. Histone modifications for open chromatin mark like H3K4 trimethylation, and closed chromatin mark like H3K27 trimethylation, but also transcription factors like POL2. The two first lanes, they're the data from Henikov. The first lane is the lane where it's encode ChIPSeq data. The second lane is the CUT&Tag with the same antibody. And you can appreciate that compared to encode ChIPSeq data, we take really nice reproducible and really nice high correlation with these marks. We try to see with a different type of cells and a different conditions. In that case, we used fresh cells and we try to go down to the number of the cells. In this experiment, the first lane is a ChIPSeq with H3K27 trimethylation. The second lane is the CUT&Tag from Henikov's lab. And then we try to go down to the number of cells. And as you can appreciate, for this mark, we can use 3 million, you can go down to 3,000 cells in order to take really nice data and comparable to Henikov's protocol and to ChIPSeq data. We try also frozen cells. And again, the same principle. We see that using 3,000 cells or even 6,000 cells at that point, we can take really nice reproducible data even though we use frozen cells. Our capabilities include the kit that I described before. We go down to 5,000 cells. There's a documentation in native conditions, and we take fast and reliable chromatin data. But also we provide end-to-end services. Here is the list of the antibodies that we have currently validated internally, but the list is growing day by day, and I can assure you now it's bigger than this list here. And I hope we will be able to validate internally all our antibodies for CUT&Tag. We request cells for the moment, roughly 50,000 cells per reaction. Soon we're going to have tissues, and the time of turnaround, the turnaround time for the full complete experiment is up to seven weeks. This I mentioned is an end-to-end experiment, including the bioinformatic analysis. Why this is all important? Because the the biggest problem of precision medicine is to have precision maps. Genetics doesn't give the answer. And here's an example between healthy twins and cancer twins that even though genetically are quite similar, epigenetically they have a lot of differences. That means in order to achieve high precision maps, you need to include also epigenetics data in your omics analysis. And here's an example for collaborations that we have with clinical labs, mainly from tumor, brain tumor, and actually tumor in prostate cancer, actually. The first example comes from a collaboration that we have in Germany, in Heidelberg, where we try to map and provide epiomics data sets to clinical laboratory in order to stratify cancer patients that they have a typical teratoid tumor. It's a really aggressive infantile tumor, and the scope of this actually protocol of this actually collaboration was to identify specific epigenetic profiles of the patient in order to design targeted therapies. We try a combination of open and closed chromatin marks, and as you can appreciate for different color codes, we are able to stratify patients and individuals and help scientists later on to design actually targeted therapies. Here is an example how you can use the epigenetics to identify novel biomarkers. A collaboration we had with a laboratory in the US, where we had to deal with FFP samples from a colon. Here is the 10-year-old samples. And the first thing that we did is to run a ChIP-Seq with H3K4 trimethylation. We are able to identify that this marker is located really nicely at the promoter region of this target gene, that's a PTK7, a known biomarker, but also in this new gene, in the promoter of this new gene in the cancer conditions. Of course, this is potentially a novel biomarker. We wanted to investigate further. We use different samples and the same marker, H3K4 trimethylation. With the ChIP-qPCR, the promoter of this region, we are able to see that enrichment of open chromatin mark of the promoter of this gene. That is potentially a biomarker for colon cancer. Really nicely, we validated with targeted bisulfite sequencing experiment where we see enriched DNA demethylation at the promoter of this gene. And now there are studies of developing drugs against the activity of this gene. Like a conclusion, I hope that I convince you that the Active Motif enables epigenetics and chromatin applications. We provide easy and efficient protocols to study chromatin and novel ways to identify biomarkers and providing epi-profiling for cancer and patient stratification. Of course, we are here to help you to choose the right tool. And with the help of Basepair, also, we are able to help you to integrate your data and to provide more complete solutions to your analysis. With this, I would like to thank the entire team for providing me all this data and all of the support. And yes, the floor is yours now, Mason. Thank
you. Okay, so thank you, Dr. Sarantis. That was an excellent talk. And we're going to move on to the second section of our webinar with Dr. Amit Sinha. He's the CEO and founder and head of bioinformatics of Basepair. His talk will focus on how Basepair has developed bioinformatic pipelines that use the next-gen sequencing data that Sarantis was talking about from techniques like CUT&Tag and ChIP-Seq to enable their users to process, analyze, and interpret data, data used using advanced analysis software. So thank you so much for being a part of this webinar, Dr. Sinha, and we're excited to hear what you have to say.
Thank you, Merson. That was a very comprehensive presentation, Sarantis. You've already set the stage for the audience. So for my part of the presentation, I'll focus more on the data analysis, especially how we enable this data analysis through Basepair. To give a very quick intro, Basepair is democratizing genomic analysis. That has been one of the big challenges in the field, that the bench scientists are the ones generating the data. But once the sequencing is completed and you have the raw data, they have to go to the bioinformatics team, and the turnaround time can be weeks, if not months, to get the results back. And in many cases, sometimes biologists have simple questions. I tried a new reagent. I tried changing step 5 of my assay. Is my data still good? Do the results look just as impressive? And it is quite annoying to wait weeks and months to get these answers. So this was our motivation to develop Basepair, so that even bench scientists without any programming skills can go from the raw data to results within a few minutes or at most a few hours. And so what we did is we built a solution from the ground up, focusing on all the challenges we typically face. So one of the challenges that these data sets are huge. So you need a lot of storage, a lot of compute. So we built Basepair completely on the cloud. So we practically have infinite storage and computing. The second challenge is the pipelines can be complex, they can be manual, they can be pretty ad hoc depending on who built these pipelines. So to solve that, we have more than 40 different pipelines, which are completely standardized, completely automated, and so on. And finally, it's all about making sense of your data. So when you do these analysis on Basepair, you get not just Excel file or static figures, but you get a completely interactive report where you don't just see the results, but you're able to change the parameter, look at the updated results, all in real time. So this is how Basepair works. Any scientist goes to our web application. They are able to upload the raw data directly in the browser. We are able to ingest hundreds of gigabytes of raw data through the browser alone. For enterprise customers, we have integrations, so we can pull the data directly from the sequencing machine or a cloud drive or so on. So we upload the data, that's step 1. Step 2 is run one of the many analysis pipeline that is the most relevant for your data type. And then within a few minutes, you get an email saying that your analysis is complete. And when you go to the Basepair website again, you see a nice interactive results for all of your data analysis. So this is really the easiest and the quickest way to analyze your ingest data. Upload your files, click a few buttons, and within an hour or so, you have all the results ready for you. We have pipelines for all types of NGS data. We have pipelines for variant calling, whole genome, exome, panels, copy number, structural variants. And then the focus of today's talk, we have pipelines for epigenetic data, ChIPSeq and CUT&Tag and so on. And even though we saw the differences between CUT&Tag and ChIPSeq, your mileage may vary depending on which type of cells you work with, which type of assays you run. So it is always a good idea to, especially in the early phases of the project, to run both of these assays and see for yourself how the data differs, which technique works better for you. So on Basepair, once you have your data uploaded, you can run any of these pipelines on your data, see how the results differ, and you can come to your own conclusion on what technique works best for you. And then, of course, one of the most popular set of pipelines we have is for transcriptomic data. So this is RNA-Seq, differential expression, gene fusions. And now recently, we have also added single cell pipelines. Currently for RNA-Seq, in the roadmap, we have a single cell for CUT&Tag and for ATAC-Seq and so on. And in addition to these standard pipelines, we also support custom pipelines. So CRISPR is very popular these days. So we have some pipelines for CRISPR analysis. We also have a pipeline for COVID variant analysis. So with 2 of our partners, we have added pipelines which can analyze COVID NGS data and tell you the variants in a given sample. So we constantly evaluate the best tools for each of these assays, and then we string together these pipelines, which are fully automated. We validate them very extensively on public data, sometimes working with customers. And when you sign up for Basepair, on day 1, you have all of these pipelines available to you. So now moving on to epigenetics-based analysis. These are some of the key steps that Sarantis alluded to. So we generally start by pre-processing the data, which means removing adapter sequences or trimming poor low quality bases. And then we use most frequently public tools that we set up on Basepair. So you get the same accurate result as you would do them by local analysis, except that with Basepair, you get the results back in a few hours and not weeks or months. So we typically use FastP. It's a tool which can trim the data for poor quality bases and also for adapter sequences and so on. The next step after trimming is alignment. This is where we take these short reads and we align it to the whole reference genome to understand the source of that specific fragment. And we use the tool Bowtie for this, another very, very popular tool for epigenetic data analysis. And then the next few steps are filtering these alignment for quality metrics, insert size, and so on. And we convert the output into a number of different formats, so they are suitable for downstream applications. And there's a bunch of utility tools for these, Sambamba, SAM tools, and so on. And next, when we have a spike-in added to our sample, we need to also calibrate the level of spike-in and then use it for normalization of the data. So the spike-in alignment is also done using Bowtie, but now to the reference, to the spike-in genome, as opposed to the reference genome. And the spike-in comes in very handy when you're expecting more of global change as opposed to just local change in a few regions. So there, the spike-in can really tell you if there's a global increase or decrease in the level of signal. And once we have calibrated the data, next is the most key step, which is the peak calling. We try to find the regions of the genome which has a high density of signal indicative of an epigenetic event. It could be a transcription factor binding, it could be histone modification. So the peak calling is the step where we identify these regions of interest. Once we have identified the regions of interest, we want to learn more about these regions. What kind of transcription factors are potentially binding at those loci? What pathways are regulated by the genes next to those peaks? So these are two of the most common post-peak calling analysis done, and we use the tool Homer for both of these steps. And finally, once we have done all of these analysis, we would want to present our data in many different ways. So a common figure is to show a heat map showing the signal density across all the peaks or a subset of peaks or genes of interest. Another common technique is to use a genome browser. So people are able to view the raw data just to make sure the data is high quality, the peaks are real, they look like a real signal as opposed to just a noisy region of the genome, and so on. So now we will go through all of these steps in detail and see how Basepair makes it very simple to look at these analysis results. So the first step we saw is data QC and pre-processing. So on the left, we see a figure showing the average base quality across the 100 base length of the reads. So we can see that the data quality is well above 30, starting from the 5' end. And even on the 3' end, the data quality remains above 30. This is indicative that your raw data quality is good. And it is very important for biologists to look at these figures because they can indicate if there has been any issues in the sequencing, where the data quality was not good or so on. And then we also do a trimming where we trim away bases which have a low quality score. And in this case, if you compare to the figure in right, you see that the base quality at the 3' end is a few points higher in the figure on the right because the poor quality bases have now been trimmed away. So it is quite common, although as you can see, the difference is small. So trimming the bases should not have any material impact on the results of your analyses. The next is understanding the whole read count flow as your data goes through this analysis pipeline. So if we start with a certain number of reads, how many of those reads actually align to the genome? How many aligned uniquely at a single position versus how many aligned to multiple positions? And then finally, you want to know what percentage of reads were duplicates. And depending on the assay, we may want to remove these duplicates. So in this case, we see that we started with about 17 million reads. In the trimming step, itself, we lost about 1% of the reads. So then we use the remaining 99% reads and align to the reference genome. And out of these reads that we aligned, about 48% aligned uniquely at a single position, and 21% of the reads aligned at more than one position. So this could be a problem for some types of analysis. You may want to exclude that, but by default, we do include these multi-aligned reads as well. So 30% of the reads did not align to the genome at all. So now we work with this 67% of the reads that aligned, and then we remove the duplicate sequences. So in this case, we see that the duplicate sequence were less than 2.2%, which is a good sign. So in the end, we have these 97.8% of the sequences which were uniquely aligned, which were a unique copy in the whole data set. So this single figure can tell you a lot about your assay. So in this case, we see the unaligned percentage is almost 30, a little bit higher than what you would expect. So this can, especially if something goes wrong, you don't see expected results, it makes sense to come back to this figure and see where exactly in the process that you're losing results. It could be if the data quality is bad, you lose reads during the trimming step. If there is any sample contamination, then many of that DNA will not align to the reference genome, and you will see very high unaligned percentage. If there is excessive PCR duplication, then the duplicate rate will be very high. So then you would want to reconsider your assay and the number of cycles you're running to reduce that number. So it is very, very advisable before you trust the downstream results is to actually look at these figures, understand the whole data analysis workflow, just to make sure the numbers are within accepted range. So the next step is, once we have this clean data, we believe it, and the numbers all look good, then we subject it to a peak calling analysis, where we are trying to find the regions where there is an excessive signal compared to the background or to the control sequences, as the case may be. So in this case, the result itself is just a table. We will look at the visualization shortly. But in this table, we see the chromosome where the peak was found, the start position, the end position. And then we have some statistics. So there's the p-value shown in -10 log scale, and then the fold change. Further, we annotate these peaks to promoter regions or intergenic or gene body and so on. And then we associate these peaks most frequently to the nearest gene, as long as the nearest gene is within 100 kb. And if your interest is in downstream finding the genes of interest, then it makes sense to find the promoter peaks, find the genes they are nearby, and then those genes are the ones which are likely to be affected by the epitope of interest. The genes annotation could be further broken down into intronic and exonic as well, as the case may be. And once we have these peaks, the next step is to find the motifs in these peaks. They could be indicative of the transcription factors which are taking advantage of these peaks. So this is relevant either you do a transcription factor assay or even a histone modification assay. If you do a transcription factor assay, the motif analysis should first of all pick up the transcription factor you're working with, because they should obviously be enriched in those peaks. And secondly, they will find, this analysis will find cofactors, which are other transcription factors which are co-binding with the transcription factor of interest. On the other hand, if you're using a histone modification assay, and in that case, let's say you're using any of the activating marks like K4ME3 and so on, then the motif analysis will show you the actual transcription factors which are binding to those regions and driving the gene expression. Or if it is a repressive mark, then the transcription factors which are potentially involved in the repression of those gene expression. So now coming back to the visualization aspects of the software, one of the things we do is look at the data in a genome browser. So even though we have a peak calling table, sometimes the statistics can be misleading, especially in noisy regions. So in this figure, we are looking at the first track in red is the treatment sample. In this case, it's GATA2. The second sample in blue is the control sample, just a genomic input. And the third sample actually shows the peaks in solid blue bars. In this example, we see that indeed 2 peaks have been found. One of the peak is on the right at the promoter region of the MYC gene. And the second peak, a much bigger one, is a little bit downstream of the MYC gene. So in this case, looking at the data, the peak appears to be very real, and the gain compared to the background also appears to be very, very real. So it always makes sense to manually look at least a few regions of interest, especially if you plan to do downstream experiments on those regions. It doesn't make sense to spend weeks or months of further time in the lab without being sure that those signals are convincing. In Basepair, these genome browsers are built in. So if you have your samples in the platform, you will be able to open up any sample of interest in the genome browser and visualize these regions, do interactive searching for genes of interest, go to chromosome markers of interest without even having to download any file at all. And the next visualization, very common with epigenetic data, is drawing these heat maps. So what these heat maps represent in the bottom panel is the signal intensity, and each row in the bottom panel is one area of interest. In this case, it's a gene. So we have about 25,000 genes and shown one in each row. And the figure on the top, the line graphs, they show the average signal across all of those 25,000 genes. And the first column on the left shows a signal all over the gene body where the length of the gene is normalized to make this figure possible. And the figure on the right is the transcription start site, zoomed into the transcription start site and looking -3 kb to +3 kb upstream. In the first figure, we see that in this case, this is a GATA2 again example, that the signal is very high at the gene start, and then it decreases as we move further in the gene body. And in fact, at the gene end site, the transcription end site, there is a small dip even before the signal returns to the background levels again. And if we look at the figure on the right, where we have zoomed into the transcription start site alone, we see a very clear localization about a few hundred bases upstream of the transcription start site, which would be what you expect with any activating transcription factor. And then we also see this little shoulder dip to the right of the transcription start site, which further shows the pattern of binding for GATA2 at these promoters. So now looking at some of the differences between ChIPSeq and cut-and-tag analyses. Overall, the process is very similar, and most of the steps we just saw, they are the same between both of these assays. The few differences are that starting with the data itself, ChIPSeq is able to work with even single-end data, whereas cut-and-tag is only recommended with paired-end data. And adapter trimming is often not recommended because we use the insert size filter, which we'll talk about in a second. And the same applies for duplicate removal. When we have ChIPSeq sequence, there is a likelihood of detecting PCR optical duplicates. So duplicate removal is a recommended step. Whereas with cut-and-tag, the way the enzymes work, you do expect the cut at exactly the same base, which would look like duplicate sequences, but that is just a reflection of the enzymes cutting the DNA at the same loci, as opposed to random shearing with ChIPSeq. So duplicate removal is often not recommended for cut-and-tag. Next, when we come to the bow tie alignment, with cut-and-tag, given our interest in finding genes bound to the protein or the histone mark, we often filter based on insert size. The most common recommended filter is between 10 to 700 base pair. So when you run these pipelines on Basepair, the bow tie parameters are already tuned for you. So all you need to do is choose the ChIPSeq or the cut-and-tag pipeline, and we will automatically handle the parameters for you. You're welcome to change these parameters and rerun your analysis with different settings, but the default parameters should work well for most use cases. Another difference we have seen is in cut-and-tag, there could be an overrepresentation of mitochondrial reads. So we recommend a special filtering step to remove the mitochondrial reads from the data set. Otherwise, these mitochondrial reads could be 20–30% of the reads and really minimize the signal coming from the remaining 70% of the real data. So it makes sense to have a filtering step where you remove these mitochondrial reads and then only do a peak calling with the remaining data sets. And coming to peak calling, MAX2 has been one of the most standard tools for epigenetic data sets developed by Shirley Liu's group at Dana-Farber. Although with cut-and-tag, the Heinkorf group itself has developed a new tool called Seeker, which has been optimized for cut-and-tag and cut-and-run type analyses. So looking at the differences between these two tools, we don't want to start a flame war, but very briefly, MAX2 has much better support for multiple samples and controls. When you run MAX2, you can easily point to multiple treatment and multiple control samples, and MAX2 is able to do the normalization internally. Whereas with Seeker, you need to do those, you need to merge your samples and controls yourself and then normalize them even before Seeker can start, start the p-calling process. Again, on Basepair, we have done all the hard work. The Seeker pipeline on Basepair automatically does merging of the samples and controls and so on. In fact, Seeker is even more sensitive to the signal in negative control and overall works much better with, where there's low background signal, whereas Max2 struggles when the background signal is quite low. So that's all for my presentation. Back to you, Mason.
Great, thanks, Amit. We're going to go ahead and move on to the FAQ section. We have quite a few questions already, so I'm going to apologize in advance if we don't happen to get to yours, but we're going to do our best. Both speakers have agreed to try and get to everything. So I'll just go in order here. So the first question, this I believe is for Sarantis regarding PATN5 transposase. Does it carry any DNA sequence of E. coli that it can, I guess, make it through into sequencing libraries?
Thank you, Spiridula, for the nice question. Actually, not the same reproducible amount. Sometimes we see quite few, sometimes we don't see at all. If your question is regards the spiking, if you can use E. coli for the spiking, I would say no, definitely not, because we cannot take the same reproducible amount, and sometimes we don't take at all for coli DNA. And I hope that I answer your question.
And then from the same viewer, he asks, how can I evaluate the duplications level? And I'm not sure if that's regarding E. coli or if that's regarding some other, it's hard to know what part of this talk he was regarding, but
yeah, from a computation perspective, the Basepair report will show you the duplication level. If you recall the flow figure, it will show you exactly what percentage of reads were found to be duplicates.
Great, thanks, Amit. Next question is, what is the DNA size after CUT&Tag? Have you run it on a gel to check? Is it similar to traditional chip sonicated fragments that are in the 200 to 600 base pair range?
Thank you, Lu, for the question. Actually, we ran the bioanalyzer before we sequence, and we compare with the libraries that we prepare for a ChIPSeq, right? We take similar size of the libraries, around 300 to 600 base pairs. And yes, it's comparable to the library preparation for the ChIPSeq. And for this, we don't lose any information also on top of that. Thank you. I hope that I answered your question. Thank you.
Regarding, it says, this question is, protein A is good for rabbit antibodies, but why don't you use protein G TN5 in CUT&Tag, which would be good for both rabbit and mouse antibodies?
Yeah, excellent question. Really, thank you again for giving this opportunity to say that we are in the process of developing also protein G. But on the other hand, we run side by side monoclonal, mouse monoclonal and rabbit polyclonals for the same targets using the protein A, and we have seen exactly the same data. Depending the target, depending the antibody, for mainly histone modifications we have tried, both of the mouse and the rabbits, they're performing the same way for CUT&Tag. But yes, we are planning also to have protein G in order also to capture more things, or maybe things that we miss with only protein A. Yeah, excellent question. Thank you. Thank you very much.
So just to try and mix up the types of questions a little bit, I'm going to skip around. So this one should be for Amit. What exactly do you mean by by $20 to $200 per sample? When would it be 200 and when would it be 20?
The price depends on the size of the data set. So Basepair can handle all the way from whole genome sequencing, which are hundreds of gigabytes of raw data, to CRISPR samples, which are not even a gigabyte. So the price depends on the type of data. The bigger data sets have a higher cost and smaller data sets have a smaller cost.
Makes sense. Next question. How does Basepair optimize parameters of peak calling for different markers or different conditions?
Yeah, we do a lot of validation with public data. We using publications from good labs. That being said, none of that is sacrosanct. When you run the Basepair pipelines, you have the option to change practically every single parameter. So you can run the same tools like MAX or Seeker, but you don't need to know R. In the Basepair user interface, there is a place to enter these parameters, which are then automatically handled by the Basepair platform to run those analyses. And you can even run the same sample multiple times with different parameters until you find something that works best for your data.
Great, thank you. So next question is, how many different transcription factors or chromatin associated proteins have we tried for CUT&Tag? Can you give us some idea of the precise experience CUT&Tag on non-histone mark antibody?
Yeah, I mean, Jeff, for sure, you know that CUT&Tag or transcription factors, there are not so many publications out there. We are aware of that, and we try to test different conditions and different antibodies. So far, we are sure for POL2, we are sure for CTCFs, and some other cohesions. We try to expand this, playing a little bit with mild fixation conditions as well, to increase the number of transcription factor antibodies that perform. But for the moment, POL2, CTCF for sure, maybe some cohesions, yes. We need to expand and we are working on that. Yes, excellent, it's an excellent point. Yes, thank you.
For CUT&Tag, they are asking for clarification. Did we say that they need to be grown, cells need to be grown on concavalent A beads, or if so, why? Or can it be done in a solution cell suspension?
Nelson, I'm sorry for this. I didn't explain myself correctly. No, you don't need to grow the cells on concavalent. Actually, you use afterwards concavalent A beads to immobilize the cells of the nuclei for doing the downstream experiment. And why you need to mobilize? You need to mobilize them because you go for the wash and you don't want to lose material. We're working with a low input amount. Now, we have tested for both in suspension and attached cells, and it performed nicely. The only thing you use acutase or scraper to remove the attached cells. You don't go harshly, for example, with trypsin, because trypsin may interfere with concavalent A binding to the nuclei of the cells. Just only this keep in mind. But yes, could work also with suspension, and could work both with suspension and attached cells. Thank you. Thank you very much.
So this one would be for Amit. What happens if somebody needs to reproduce an analysis outside of your platform? Can pipelines be exported and run outside of the Basepair ecosystem?
Yeah, when you run analysis on the Basepair, the exact log of every single tool is generated automatically. So you're able to download those log files. You can see which tools were used. You can see exactly which parameters are reused. So you can run the same analysis on your local machine as well. Great.
And another one for you, Amit. It says, first of all, thank you for a wonderful presentation, and I agree on that one. It was stated that to do QC, that you should do QC before and after trimming. What if before the trim, the QC is less than 30? Would you continue, or what would you do in that case?
Yeah, that's a good question. You can continue because with ChIPSeq, the goal is not to find a very precise alignment. The goal is just to find the region of interest. So even if a few bases have low quality, the reads will still align to the right place in the genome, and you know that there is a signal coming from that region. So as long as the reads are above 10 or 15 base quality, you should be able to go ahead with the data, even though the ideal is 30.
So another question from Nirmal Kananian. He's interested in, or they are interested in transcription factors, and some of these do not have great antibodies for how they want to study them. So they're considering using different tags like HA or flag tag. And the question is, do you think antibodies against these tags can be used with CUT&Tag?
Yes, excellent question, Nirmal. Thank you very much. I mean, yes, we are trying to validate internally. Of course, depends the target. And of course, depends how well it's expressed in the cell, right? If you consider a target that is really close binding to the chromatin and is quite highly expressed, then has a lot of chances to perform nicely also with CUT&Tag. But it's something that we are currently validating, and soon I will have more information to give you on that. And if you send me your email also, I can give you some updates from our side. Thank you very much for asking.
We should, I believe when they registered, we have their email, so you can follow up, Sarantis. When,
yes, super, that would be great. Yes, that would be great. Yeah, yeah, super. Thank you.
So another question that I think sort of relates to that is, or just by matter of clarification is, can you explain the differences between CUT&Tag and cut-and-run? And can the CUT&Tag be used for transcription factors? You talked a little bit about that already, but yeah, I think that's a common question. So
yeah, it's a common question. Mainly is the use of MNAs, both of them native conditions, CUT&run use the MNAs, CUT&Tag use the transposon. This is one of the major differences, and the integration of the sequencing adapters for making the library. In regards to transcription factors, CUT&Tag can be used. Again, depend the transcription factor, depend the target, and depends the antibody. I would not say there are not so many publications, of course, still for transcription factors, but I'm guessing slowly people, they get into that. And we are internally working on that, trying to optimize a little bit the protocol by integrating and putting a little bit of fixation, for example. This hopefully will give us more, let's say, possibilities of picking up of transcription factors, more transcription factors. Yes, that I hope that I answer your question, Alex. Thank you.
This one will go back to Amit from Daihu. What do you do when the duplication rate is very high?
Yeah, that's a tough one. Depends on, is it even possible for you to, let's say the duplication rate is too high, and is it even feasible for you to rerun the assay again? If you only have limited sample to begin with, and you're no longer in a position to rerun the assay, you may no longer have a choice but to just work with the data. You may throw away the duplicated reads and keep the unique ones and see if the results make sense. It's always good to have a few positive and negative controls, a few genes, you know if they behave in one direction or the other. And if your positive and negative control genes are showing the right pattern in terms of gain or loss of these marks, then you may work with the data. It's always ideal to run the analysis again. And bioinformatics people always ask for it, even though on the lab side, it may not be just possible to duplicate the assay again.
And another bioinformatics question, what type of negative controls would be required for seeker peak calling?
We have tried both the genomic input and the IgG controls.
Oh, quick one.
And sometimes if you're comparing two different conditions, then the background is presumably canceled between the two samples, and you can compare two different marks directly to each other.
Excellent. A quick question about, I'm not sure, actually I might, so this question is from Sarantis. When you mentioned using frozen cells, I'm not sure which exact.
Cryopreserved, yeah, D. Thank you for asking this. I'm sorry if I didn't explain correctly. I mean cryopreserved, not snap frozen. Cryopreserved cells.
Yeah. So this one is from Ruikeng. How do these two protocols work on proteins that are indirectly associated with DNA, like chromatin remodelers? Would cross-linking be required in those cases?
Ruikeng, yes, I will say yes. Cross-link will be required, and for ChIPSeq, obviously, for CUT&Tag, maybe a mild also fixation may require for proteins that are not tightly or close correlated to the chromatin. Yeah, I agree with you.
Yeah, I've also heard of people using light fixation, or they've been playing with it to try and get better for transcription factors as well.
Yeah, that's a good point.
I believe this is the same viewer. When doing cut and run, the beads are heavily aggregated. How can they improve this, and does it affect the results? I have some ideas, Sarantis,
yeah, I mean, feel free, Mason, of course, feel free, please.
Yeah, so I've noticed that sometimes this can be affected if the beads have been frozen, they'll clump more. You should avoid freezing, but there is always a little bit of clumpiness, and you can break that up with a pipette tip or other agitation, and it can affect results if the beads are clumping up. Then obviously they're not going to be able to access, where they're supposed to with your sample. So yeah, a little bit of aggregation is to be expected, but too much is not great.
Confirm.
Did I? If there's anything else you have to add, that would be good. Okay.
No, perfect. You covered completely. Thank you. Great.
Let's see. I want to make sure we're getting a good mix of questions here, so I'm going to skip around. This one's a quick pricing question for Amit. Does the cost per sample of uploading data at Basepair, does that include analysis?
Yes, it does. And in fact, it includes unlimited analysis. So if you have multiple samples, you only pay for the samples, and you can compare them to each other as many times as you like. And if you want to test different parameters, again, you can run them as many times as you need. There's no extra cost for those.
Great. Again, so another one, looking at the genome visualization, there's a peak not considered in the analysis. Is it possible to include manually some peaks that are considered bona fide peaks?
Yeah, I think manually adding the peak will make the reviewers very unhappy if you're trying to publish the results, because peak for one is not the peak for the other. So the reason the other may not have been picked as a peak is maybe the cutoff we have for the peak calling, which is the default is P 10 to the power -5. So if your region, and that is exactly, it's a great question, because this is exactly why you should not just depend on the statistical result, but look at the regions of interest manually, at least a few of them. In this case, if you see a region of interest where you see a peak, it looks believable to you, but it is not getting picked up, you would run the whole pipeline again, but with a lower p-value threshold, let's say 10 to the power -4. And then with this lower threshold, the peak may be picked up. And obviously the downside is you may pick up too many peaks now all over the genome. So that is a fine tuning you need to do, that the p-value should be stringent enough to pick up good peaks, but not pick too many random bumps all over the genome. But yeah, so I would not recommend manually picking the peaks, but maybe running with different parameters, which would pick the peaks of your interest.
Great, thank you. So I'm seeing a couple questions that from a few different people asked in slightly different ways. So I think I'll take this one and make it our last question. I apologize to anybody who didn't get their question answered. We will follow up individually with you. So this question has to do with cut and tag controls and spike-ins. So this one's kind of a multi-part question. First one is, does the IgG negative control need to be sequenced? In this person's case, they've been getting no peaks on their bioanalyzer for the IgG, and they're wondering if that's a problem.
Yeah, I can tell you for our service and for our experience, and please, Mason, correct me if I'm wrong, we strongly recommend it, and we do it, to run the IgG. This is for the stickiness of the antibody and, you know, the things that you control. This is our negative control. Now, I don't know why you don't take sequences or how much recovers you are taking in the sequencer, but please feel free to drop us an email. We can discuss on that. Now, in regards to the spiking control, it would have been great to have a spiking control. So far, to my knowledge, there's no something really well established. Recently, I saw a paper for a protocol describing a synthetic DNA based on an ampicillin gene that they use it for during the library preparation to normalize kind of spiking. But this is the recent advantage that I see. I haven't seen something else. We are currently working of developing our own spiking. It would have been great, and we are really looking forward to have it. For sure, we will communicate it to the community once we have managed to have really reproducible data with our spiking controls. Are they both of the above controls required for peak calling, and how much excluding IgG and spiking will affect the final data quality? This probably Amit will answer this part of question. I would say rather than exclude it, help it to equalize and to normalize, especially for the spiking. But Amit, maybe you can add something from your.
Yeah. So yeah, that's exactly right. The genome is not unbiased. There are certain parts of the genome which end up showing more reads, even if you just sequence blank input genome without any selection. So when we do peak calling, we see the gain of signal compared to this IgG background.
Yeah, yes.
And how much above the background it needs to be, that depends on the p-value and the fold chain thresholds you have.
Absolutely.
Yeah, so I think we've only gotten through about half the questions, and they are all really good questions. So I'm going to ask Amit and Sarantis to follow up and answer these individually. Sarantis, if you could put up the last slide, please.
Yes, yes, I'm sorry.
But I wanted to thank everybody for tuning in. If you have any further questions or would like to get in touch with either one of us at Basepair or at Active Motif, you can visit our websites, activemotif.com or at basepair.com, basepairtech.com, and use the email addresses that you see on the screen. And on behalf of everybody at Active Motif and at Basepair, I'd like to thank you all for your time and your attention, and we wish you all the best of luck in your research. Have a great day.
Thank you very much. Thank you.
Thank you so much, Mason. Sarantis, a great talk as always.